Good morning. Okay, same protocol, door shut, empty room. Hang on just one moment. Let me adjust this camera a little bit. Here we go. And here we go. Chapter 2, Chemistry of Life. And my clicker's not wanting to work today. Okay. So, first chapter, we talked about life, what it consists of, that biology is the study of life. Life's made up of cells. Cells are made up of molecules. Molecules are made up of atoms. Then we have molecules. We have things like proteins, and we're going to talk a little bit about proteins today. We have enzymes, which are proteins, and there's carbohydrates, ATP, and other things as well. Still trying to get my clicker to work. We're just not going to have there we go. This is a periodic chart. This is actually only a partial chart. It's actually got a couple of more rows on it here. These are the ones that are important for biology. And if you look at this little chart right here, these are the ones that are extra important. Basically, the only ones you'll be responsible for are oxygen, carbon, hydrogen, and nitrogen. Later on in the chapter or later on in the semester, we may talk about some of these trace elements a little bit. All molecules or atoms are made up of pretty much the same way. The atom is the fundamental unit of matter. Atoms are going to contain protons and neutrons in a structure called the nucleus, which is sort of centrally located. And then around the nucleus, there are the electrons. The neutrons have a neutral electric charge. They do not have a charge at all. The protons have a positive electric charge. The electrons a negative electrical charge. And since we have one item that has a positive charge, one item that has an electrical charge, and for stability's sake, those need to be equal, we can always determine that in its natural state, an atom or an atom of an element will have the same number of protons and electrons. Again, that's sort of a chart showing what I just showed. The electrons negatively charged. The electrons are incredibly small. We're going to talk in just a second about something called the atomic mass. The atomic mass is figured, or it's calculated by adding the numbers of protons and neutrons in the nucleus. Since the electrons are so small, even in some of these very large elements where there's maybe dozens or maybe 100 or more electrons, we don't include the electrons in the atomic mass simply because they're so small. The neutrons have a mass unit of 1. Protons have a mass unit of 1. So to calculate the atomic mass of a particular element, you would simply add the numbers of protons and neutrons. On the left is a neutral hydrogen atom. You see this has 1 proton. The green dot represents the proton. There are no neutrons in a typical hydrogen atom. There is 1 electron. So you have 1 positive charge, 1 negative charge for a net charge of 0. However, what can sometimes happen, especially after molecules have formed and then bonds break, atoms may be gained or lost. And we'll talk about the gain or loss of electrons in just a second. But when an electron is gained or lost, we now have different numbers. And this one we're showing you right here is just a hydrogen ion. The electron has disappeared. It's gone away. It's been lost through a reaction. We now have 1 proton. We no longer have an electron. So we now have a positive 1 charge. And anytime you have a charged particle, those things are referred to as ions. Again, just another artist's rendition of a typical atom of some item. Again, larger atoms are going to have more protons and neutrons. And there's always, if you take the time to count, you'll see there's 1, 2, 3, 4, 5, 6 electrons and 1, 2, 3, 4, 5, 6 protons. Some other typical ways that atoms appear is in what are called isotopes. An isotope is simply a form of an atom that has different numbers of neutrons. Same numbers of protons, same numbers of electrons, but the neutron number is different. And the one we show here, the one we typically talk about, is carbon. Carbon comes in three forms. There's carbon 12, which is the most common form. You have 6 protons, 6 neutrons, and 6 electrons. So 6 neutrons plus 6 protons equals 12. So the atomic mass or the atomic weight of carbon 12 is 12. Then you have another isotope of carbon, which is carbon 13. You have 6 protons, but now you have 7 neutrons, but still 6 electrons. Keep the protons and electron number equal. Therefore, you have a stable non-ion form. And then finally, there's carbon 14. You may have at some point in your education, or just watching television, you may have heard something referred to as carbon 14 dating. That's what we're doing. We're looking at the amount of carbon 14 that's in an organic substance. Still have the 6 protons and 6 electrons, but now you have 8 neutrons. And if you look at the typical little block in a periodic chart, you'll see the atomic number. The atomic number always refers to the number of protons in a single atom of that element. You will then see the element name. Some charts, it will be written out. Other charts, it will not. All charts will contain the atomic symbol, which is a 1 or 2, or in some rare cases, 3-letter abbreviation for what that particular element is. In this case, C means carbon. And then the atomic weight, you see the atomic weight of carbon is 12.0112. The way that is calculated is the atomic weight of this one is 12, the atomic weight of this one is 13, the atomic weight of this one is 14, and if you add all of those up and take the average of this one plus this one plus this one, you get 12.0112. There are some other typical elements showing what is called the valence shell. Again, the electrons appear in these structures that have had many names throughout history. They've been called orbitals, they've been called clouds, they've been called shells. Your textbook uses the word shell. All elements have these. All but 2 elements have multiple shells. Hydrogen and helium are kind of unique in the fact that they only have 1 electron shell. And that first electron shell, the one that is closest to the nucleus, no matter which one you're talking about, will hold at most 2 electrons. The little purple dots represent the electrons. So you have, here's 1 electron and 1 proton, but here you have 6 protons, so you have to have 6 electrons. Well, you have 2 in this first shell, and then you have this second shell, which in this case has 1, 2, 3, 4, has 4 electrons. Now that outer shell is referred to as the valence level or the valence shell. Any empty dots here represented by the valence in the energy shell, which is a vacancy. There could be one there, but there's not. The elements that have these openings in that outside shell are the ones that are going to readily form bonds. They're going to form bonds very easily with other elements. Things that have a full outer shell tend not to form bonds. In this case, we're going to look at where? Neon. Neon, you see it has a full outer shell. There's no open circles in neon. So neon is an element that we refer to as a stable element. There are several of these. They're called the inert or the noble gases. Things like helium, which has a full outer shell, even though it only has 2. And then there's helium, there's argon, there's krypton, there's neon, there's xenon, there's radon, all of those on gases. All of them have a full outer shell. Now, that outer shell, except for the first one, all of the rest of these outer shells are going to hold a maximum of 8 electrons. Now, when you take a chemistry class and you get into some of the larger elements, you're going to find that, yes, there are energy shells that hold 18, and I think there's one that holds 32. I don't remember my basic chemistry. It's been a long time since I took it. But that is something that we need to keep in mind, that the magic number is 8. It's so important that there's actually something called the octet rule. And the octet rule says that for an element to be stable, it must have 8 electrons in its outer shell. And again, when you get into those bigger ones, yeah, there are some sub-levels that may have more than 8, but the very farthest one from the nucleus, the outside one, or the valence level, maximum number is 8. This is a scale of what is called electronegativity. When you look at the electronegativity, that tells us how strong the pull on electrons is. So these elements are going to very likely form bonds. Here's sodium over here. Here's chlorine over here. You see chlorine is much more electronegative than sodium. So when sodium and chlorine come together, those valence electrons will literally change places, because the chlorine has such electronegativity, it's actually pulling those electrons off of the sodium. The movement of electrons leads us directly into a discussion of what are called chemical bonds. The way elements stick together to form everything on the planet is through the actions of electrons forming bonds. There are basically 3 types of bonds we're going to talk about. We're going to talk about the ionic bonds. The thing you need to remember about ionic bonds is that one or more electrons actually change place. This description says that one atom donates one or more electrons, but these electrons actually move. They go from being around element A to being around element B. They literally change place. And by changing place, they're going to disrupt that even number of protons and electrons, resulting in a charged particle. The next type of bond is something called a covalent bond. A covalent bond is one that where electrons don't change place, they're just simply going to be shared. And then finally, the hydrogen bond, it is a relatively weak bond, and it actually forms by the interplay of what are referred to as polar molecules. And we'll talk about polar molecules in just a moment. But whereas these top 2 are the interplay of electrons, hydrogen bonds really don't have electrons moving around. What they have is the positive end of one molecule and the negative end of another molecule are attracted to one another. We know opposite charges attract, and those are attracted to each other, and that's how the bond is formed. The thing about hydrogen bonds is they are relatively weak bonds, and they usually appear in really large numbers. There's usually lots of them. This is a demonstration of what happens when sodium and chlorine come together to make salt. If you look on the left-hand side, you see the sodium atom with its 1 valence electron. There's 1 electron in that outer shell. You see chlorine, if you count the dots, has 7 in its outer shell. So if sodium donates 1 electron to chlorine, then the sodium atom has 8 electrons in its outer shell. The chlorine atom has 8 electrons in its outer shell, as seen right here. However, now the sodium has a slightly positive charge, the chlorine has a slightly negative charge, and we make a product called salt. There are some very common other items. Now these are produced by something called a covalent bond. In the case of sodium, the electron is literally changing places. In the case of these items, however, the electrons aren't changing places, they're just sharing with each other. This first product is called methane. Methane is a gas. It is the simplest of the organic molecules. If you ever have to take a course in your life called organic chemistry, you have my deepest sympathies. This is the study of organic molecules, and the simplest of the organic molecules is the gas called methane. Organic molecules are made up of carbon and hydrogen. And as they get more and more advanced, but their backbone is carbon and hydrogen, and that is methane. You see, you have one carbon. Carbon needs 4 electrons to get to that magic number of 8. Hydrogen has 1. So if you've got 1, 2, 3, 4 hydrogens, 4 hydrogens can combine with 1 carbon to form this molecule called methane. And you see, instead of drawing it this way, where the artist has actually eliminated this 3rd shell from the sodium and to show the electron is actually moved, what we do here is we continue to show the valence shell, but we show where the 2 shells come together, both of the electrons. So these electrons are being shared. The second element being shown there, or excuse me, the second compound being shown there is water. You see, you have 1 oxygen, 2 hydrogens. Oxygen has 6 outer shell electrons, so it needs 2 more. 2 hydrogens, so you got H2O, H2O, and you see there shows that they are being shared. And then the last one, oxygen gas. Atoms of oxygen can actually bind to each other to form oxygen gas. This is molecular oxygen. And as again you see, the electrons being shared are shown here in sort of an overlapping molecule or overlapping orbital. When carbon forms bonds and these organic molecules start to be made, carbon is a kind of a unique element. Anybody who's ever watched the old 1960s television series Star Trek, for instance, knows that humans are carbon-based life forms. All life on this planet that we know of is carbon-based. And carbon is kind of unique. Carbon always forms 4 bonds, and it always forms 4 covalent bonds. So carbon will bond with a lot of different things, but it's always going to form 4 bonds, and it's always all bonds are always going to be covalent. And in covalent bonds, the electrons are being shared. So here's a typical, what is referred to as a single bond, where you see all of the carbons have 1 line attaching them to something else. And when you see this chemical shorthand, the line between elements represents 2 electrons forming a bond of some sort. So you've got this carbon forming single bonds. But if you count the number, it's still 4. This is a carbon-to-carbon double bond. Sometimes the electrons will be shared between the carbons. And so, but if you count, this carbon has 1, 2, 3, 4 bonds on it, and this carbon has 1, 2, 3, 4 bonds. But that is a carbon-to-carbon double bond. And then you have the triple bond, where you're having 3 pairs of electrons being shared. But again, do the counting. You have 1, 2, 3, 4, 1, 2, 3, 4. All of the carbons are following that rule of carbon has to form 4 bonds. And then, for biological purposes, especially when we start talking about things like proteins and carbohydrates and lipids, we have what are called ring structures. That's where these are all linear. They're all drawn in a straight line. These are ring structures. But again, if you look at the carbons, you see 1, 2, 3, 4. Everywhere you look, the carbon is always forming 4 bonds. This is a demonstration of water. Water is a very unique compound. It is the basis of life. The Mars rover just landed the other day during Snowmageddon 2021. And one of the things they're going to look for is the presence of water. Was water ever there? Is water there now? Water is actually unique in the fact that each individual water molecule is held together by what is referred to as a polar covalent bond. And in a polar covalent bond, the electrons are being shared, but they're being shared unequally. One of the members of the bond, one of the elements in the bond, is so much more electronegative than the other that it pulls on those electrons more, and it makes them spend more time around that element. So what happens when water forms this molecule, again, these polar, oxygen, the way this slide says, hogs the electrons. So what you wind up with is a molecule that has a slightly negative end and a slightly positive end. So what happens is, this is molecular water. But when you start looking at water forming a compound, what happens is hydrogen bonds form between the positives on one end of the molecule and the negatives on the other end of another molecule. And if you look at this picture, the dotted lines represent the hydrogen bonds. And if you look very closely at this image, which is in your textbook, you will notice that there's lots of places where the negative end of a molecule or the positive end of a molecule is not attached. It's not forming a bond. And that's why water has some of its very unique properties. The water in this bottle is being held together by these hydrogen bonds. But there's still lots of other places where bonds could form. And that's how we wind up with water having some of its very unique solvent qualities. Lots of things will dissolve in water. Water cycle. Water evaporates through the pores in leaves and goes into the atmosphere. The water will collect in the atmosphere. Eventually, enough of that water will get together and clouds will start to form. Clouds will start to form and collect even more and more water. And eventually, there's so much water in the clouds can't hold the water in its vapor form anymore. The water will begin to condense, and then it falls down to earth in what we call rain. The water then goes in through the roots of the trees, and it is pulled up through a process called cohesion, which we'll talk about in just a moment. That's how it's pulled up through the tree and out into the atmosphere. So rain falls down, the tree picks it up, goes back into the atmosphere. That's the water cycle. When you put salt in water, everybody knows what happens is the salt dissolves. Now, why does the salt dissolve? The salt dissolves because the available positive and negatives in the water grab onto the positive and negatives in the salt and literally pull the salt molecules apart. The salt begins to separate and form, again, here we call these things ions. There's the sodium ion that forms there and the chlorine ion that forms there. One interesting thing about salt. One other thing about salt is that sodium and chlorine in their elemental form are very volatile items. Sodium is an explosive metal. If you take pure sodium and you get it wet, it explodes. It will explode in water. There's a YouTube video that you can watch where a guy takes a big lump of sodium and he throws it into a pond and it hits the water and there's a big explosion and it bounces and it explodes again and again and again and again. Chlorine is a sort of a yellowish gas, has sort of a pungent odor. If you've ever seen any old documentaries about World War I and you've heard about something called mustard gas contains a lot of chlorine. It's a poisonous gas. So you take a poisonous gas and explosive metal, combine them chemically, and you make the stuff we put on our French fries. There's water molecules, and you can see in this shot, you see all these available bond spaces. As you cool things down, as you cool water down, more and more bonds will start to form, and eventually the water will start to form this nice symmetrical sort of lattice pattern and form these nice hexagon ring structures. And as more and more of these ring structures form, eventually you form ice. So you go from liquid water to ice. And if you go the other way and you start to warm the water up, what happens is more and more of these bonds will break, more of these little molecules will become individual, and you create what is called steam. Some characteristics of water that make it such a wonderful item for life on this planet. Water moves through the plant's roots to the leaves through a process called cohesion and adhesion. This simply means that water molecules tend to want to stick together. And as one molecule is being drawn up the leaf or the stem or the trunk of the tree, it pulls more water molecules with it. Excuse me. Because of all of those available bond sites, water dissolves things very readily. Water is a very good solvent, we refer to it. Water regulates temperature very well. How do we control our body temperature? We sweat. Takes a lot of heat to change the temperature of water. So if you've got a swimming pool in your yard, you know that, first part of the summer, first part of the late spring, when the air temperature starts to get really warm, the pool is still pretty cold because it takes a lot of energy to warm that water up. Water expands as it freezes. If anybody has ever had a bottle of water in their freezer or put it in the refrigerator and it freezes, you'll notice that the little dimples on the bottom of the little bottle that make it sit up, those little dimples will flare out and the bottle won't stand up anymore because water expands as it freezes. It's very chemically reactant, produces products and reactants. Therefore, the photosynthetic reactions require water molecules for photosynthesis to occur. Also, something we measure when it comes to water solutions, or what we refer to as aqueous solutions, is something called the pH scale. Now, if you've ever gone and had blood work done because you're not feeling well, you're trying to figure out what's wrong, one of the things they'll do is they'll check your blood pH. If you look right here, here's your blood. Blood should have a pH of around 7.35 to 7.45. Things that have a pH above 7, we refer to those as bases. Another word for that is alkalis. Things that have a pH below 7 is referred to as an acid. You can look at some very common things here. Rain has a pH of about 5.7. That's what we mean when we talk about acid rain. Here's cola, beer, and orange juice up around 3. Hydrochloric acid, the battery acid that's in your car is a hydrochloric acid or a sulfuric acid, both very high pHs. Strong bases, things like ammonia and household bleach. You also have something called sodium hydroxide. That is a product that is very, very caustic, but it's a very strong base. Some other functional groups that you'll need to familiarize yourself with for the biochem, something called the hydroxyl group, which is simply an oxygen and a hydrogen written in this OH form. The carboxyl group, which is shown, this is the way it chemically looks. This is the way it's typically written out. The amino group, when we talk about amino acids, this is the thing that's added to make it an amino acid later on in the semester when we talk about digesting, or if you take a nutrition class and you're talking about digesting proteins. The first thing we do to process a protein is remove the amino groups from all the amino acids. And the amino group is NH2. Phosphate group, we'll talk a lot about the phosphate group here in a little while when we start talking about the ADP-ATP cycle. It's one of these. The difference between ATP and ADP is one of these. ADP, D as in dog, has 2. ATP, T as in Tom, has 3. When we start making large molecules, we have these 2 terms that you need to be familiar with called monomers and polymers. A monomer is a single unit. Now, don't get the false idea that because something is a monomer, that means it's small. Because there are some monomer units that are exceedingly large. But if it is one single unit, it would be a monomer. You put a bunch of monomers together, here's a single monomer and another single monomer, through a chemical reaction, you add them, you wind up with a two. See, these are now chemically combined, but when that happens, water is released. That is called dehydration synthesis. You're dehydrated. Water's been lost. So through the chemical process of putting this together with this, we have a byproduct of water. Now, there's another method, or the opposite of that is called hydrolysis. So dehydration synthesis, joining monomers into polymers with the result of water as a byproduct. Hydrolysis, on the other hand, is you take the polymer, break it down into the individual monomers, but that requires water. Water is what's actually causing the bond to break. So you wind up with, if you'll notice, you have an OH and an H here, but you don't have those anymore because the water's been released. Here the water comes back in. Now this monomer has its OH on this end, and this monomer has its H on that end. So dehydration synthesis, making something, putting things together with water as a byproduct. Hydrolysis, using water to break things apart. And there are some classic examples of wonderful ideas of things that we have. Carbohydrates, showing the potatoes, the fries, proteins and lipids, the fried chicken that looks like a wing. Well, anybody want to eat a single chicken wing? I don't understand. Eat a bigger piece of chicken. And then you have carbohydrates and cellulose. Macromolecules of life. We're going to talk about carbohydrates and proteins, nucleic acids, and lipids. Carbohydrates are the simple sugars and the complex sugars. The simple sugars are the monosaccharides and disaccharides. Table sugar, the thing that you put in your coffee, those kinds of things, those are simple sugars. The complex carbohydrates, or the polysaccharides, are things like starch, glucose, glycogen, and a product called chitin. These are simple structures. These are very complex. These are very large polymers. Proteins are polymers of amino acids. We'll talk here a little bit about the production of proteins. Nucleic acids. The primary nucleic acids that we talk about in a biology class are DNA and RNA. Those aren't the only nucleic acids that exist, but those are the primary ones that we're going to talk about. And then lipids. When we think about lipids, the common term for lipids is fats. And we'll talk about several different types of lipids. So again, the monosaccharides, such as ribose and glucose and fructose, those are simple sugars. Disaccharides are also considered simple sugars. You take one glucose and one fructose, you put them together with the loss of water, and you make a sugar called sucrose. Your table sugar, the sugar you buy in the 4 and 5 pound bags at the Walmart or the Kroger or wherever you go do your grocery shopping, that is primarily sucrose. Whoops, forgot polysaccharides. And we have the polysaccharides. The thing I want to point out about the polysaccharide is look at the molecule. Look at cellulose, how closely these things are packed together. And then with starch, you see it start to be spread out a little bit. And then with glycogen, it's got a different structure to it. These different structures affect the way those things have to be processed. Human beings, for instance, we don't digest cellulose very well. We need it in our diet. As a matter of fact, we have to have a certain level of it in our diet, but we don't digest it very well. Starch, on the other hand, and glycogen, we can break those down relatively easily. Amino acids, excuse me. Again, these are the different amino groups. You see, if you'll notice, whether you're talking about glycine or cysteine or tryptophan, you notice you always see this amine group added to it. And you'll notice that this is the same. You'll notice the only thing different on these amino acids is what's referred to as the R group. What is that? What is in that position that makes the different amino acids? You notice glycine, very simple, simply has a hydrogen there. And then we have the cysteine, which has a little bit more complex. And then you get a bit of tryptophan, and look how complex that is. Yeah, I would never ask you to draw that. Your chemistry instructor might, I won't. But again, these are amino acids. And then you can show the breakdown or the formation depending on which one you're doing. Again, dehydration synthesis, you put this amino acid together with this one to make this dipeptide. You've lost water, but then you bring water back into it and you can separate them back out again. And then there are some very common forms of proteins. You have fish and red meat and chicken and eggs, very high protein in eggs. The white part of the egg is almost all protein. And then you have certain tree nuts that also contain lots of protein. And then those are supposed to, I think those are supposed to be a bean or a legume of some kind that has protein in it. Proteins come in a lot of different forms. We have what are called structural proteins that build things. Your muscles, for instance, made up of a lot of structural proteins. Antibodies, a lot of talk in the news media now about antibodies and people getting the COVID-19 shot, and their bodies making antibodies to fight the disease or the virus. Those antibodies that you are forming are a type of protein. You have contractile proteins. These are the ones that are found, again, in muscle tissue. These are what give our muscles the ability to expand and contract. And then when we talk about the cell later on in a later chapter, we'll talk about membrane proteins. We'll talk about some digestive enzymes. Here together, the ovalbumin, which is the egg protein. So that's a storage protein. You have enzymes and you have transport proteins. Molecules or proteins can be disrupted. You can get them into a high heat environment, a high acidity environment. If they're outside of their norm, they can be disrupted, their shape can be altered, and that is referred to as being denatured. So here's a nice little drawing. These are the egg proteins. You see they're coiled, they're twisted, they're going one direction or another, and then you heat them and they all sort of unfold and untwist and take on this nice little straight shape. You fry the egg. This is a denature. Anytime you change the shape of a protein, anytime you cook anything, you fry an egg, you grill a steak, whatever you're doing, you're denaturing those proteins. You're changing their shape. Excuse me. That's the reason the texture of chicken or the texture of steak or the texture of hamburger, it changes as you cook it. That's because those proteins are changing. You're denaturing those proteins. These are the nucleic acids, DNA and RNA. There are these adenine, cysteine, cytosine, guanine, thymine, uracil. These are something referred to as nitrogenous bases, and nitrogenous simply means they contain nitrogen. You see nitrogen here and here. You see the nitrogen all scattered throughout those molecules. Excuse me. But these are different bases. The difference between DNA and RNA is this sugar molecule. If the sugar is deoxyribose, you have DNA. If the sugar is simply ribose sugar, you have RNA. And again, this is the monomer. You see, it's a very large molecule, but it is still a monomer. But you put two of them together, and you make a polymer, or two or more of them together. And then there's the DNA showing, here's the DNA in the nucleus. forming either another strand of DNA to make a new cell or a strand of RNA to make a protein. Triglycerides, lipids, and fats. All lipids contain two things. They contain a glycerol, which is a product made from glycerin, which is a type of sugar. And then they have these long tail-like structures, which are actually called fatty acid tails. And you will notice on this particular one that there are three of them. Three fatty acid tails equals what is referred to as a triglyceride. 3 fatty acid tails and glycerin. Now you'll notice that these over here, these two, all of the carbons are forming single bonds and all of the carbons are surrounded by hydrogens. That is what we refer to as a fatty acid. And that is a saturated fat. If all the carbons are forming single bonds, that's a saturated fat. However, if some of the carbons, as in the case of here and here, are forming double bonds, that is an unsaturated fat. The word saturated means full. So this one is full. It's holding all the hydrogens it could possibly hold. It's saturated with hydrogen. This one, on the other hand, is missing some hydrogens. There could be hydrogens in other places, except for the fact that we've got these double bonds. So when that happens, it is referred to as an unsaturated fat. So you take this fatty acid tail, this fatty acid tail, this fatty acid tail, this glycerin, combine them together with the loss of 3 water molecules, and you wind up with this triglyceride. And this is an unsaturated fat. Different types of fats come in different areas. For instance, the old-fashioned saturated fat, the one that's been around the longest, is solid at room temperature. And the reason it is solid at room temperature is, if you'll notice how all of these tails are nice and straight, which allows one molecule to sit right up next to another, which right up next to another, and another, and another. You wind up with a very solid structure. Unsaturated fats, however, are, excuse me, they are bent, twisted, so they don't come together so easily, so they are liquid at room temperature. And the one that is talked about the most, especially if you take a nutrition class when you talk about fats, is the trans fat. Now you will notice that trans fats are drawn here as being straight, as looking like this. But look at the difference here. There's a double bond there. Now, let's back up one slide. And you see the double bond here and here? The reason these fatty acids are bent is because the double bond, at the double bond site, both of these hydrogens are on the same side. That is referred to as a cis conformation. And that cis conformation is what puts the bend in the molecule. But now let's look at this trans fat. What's happening here? On the trans fat, the hydrogens are on opposite sides. By being on opposite sides, you get this nice straight structure. Thing about trans fats is they don't exist in nature. These are fats that human beings have made. We have taken a fat and we've chemically made it change its conformation to go into the trans conformation. Cis means same, trans means opposite. So these look like this. They have this difference. And what we now know is that while these are bad for us, these are even worse. There are some sugar combinations, or excuse me, some sugar substitutes The one that's been around the longest is saccharin. Saccharin has been around since I want to say probably the 1950s. It was taken off of the market for a while because it was thought that it was a major cancer causer. It's now been proven that it's not as bad as we originally thought, so it's back on the market. We have Equal, which is another chemically produced sweetener, and then we have something called Splenda, which is the newest, one of the newer, the newest product that is supposedly a sweetener that diabetics, for instance, can eat without a whole lot of problem, is something called stevia. Stevia is a naturally occurring sugar. It's made from the leaves of the stevia plant. You can actually buy stevia plants at Home Depot and grow your own stevia leaves. Wife and I tried that. Extracting the sugar is really, really difficult. You can't do it in your home, so I don't recommend it, but it was a cute little thing to do. But again, those are artificial sweeteners. This is an artificial fat that was made in the 80s to hopefully we could start frying foods in a fat-free method. The product is called Olestra. Olestra was a fat that was designed for the cooking industry. It was used to fry all kinds of things. There's fried chicken. Again, staple meal of the South, fried chicken. But how do you fry it? What do you fry it in? Do you fry it in saturated fats? Do you take a big old scoop of lard and put it in there? Or do you use peanut oil or olive oil? What do you use? Different fats have different properties. There are good ways to fry, and there are bad ways to fry, and there are not so bad ways to fry. Some other lipids, cholesterol. Cholesterol has a somewhat undeserved bad reputation. Cholesterol is a product we need. We have to have it. We have to have a certain amount of it. It is a product that we use every day. Every time we make a new cell, for instance, we have to have some cholesterol for that new cell. Testosterone, again, if you'll notice, this molecule here, just take a sort of a cursory look at the molecule and look at this part of this molecule right here. You'll see it's very similar. What the body actually does in men is we take this molecule, cholesterol, and we cut some things off of it and we convert it into that. One of the reasons you have to have a good LDL to HDL relationship, especially if you're a male, is for proper testosterone. If you're really trying to control your cholesterol and your cholesterol number goes down, a subsequent problem with that could be low testosterone. If you have low testosterone as a man, it may simply be that your cholesterol levels are out of balance. They should not be where they are. Fat reading labels. Again, anything you need to know about labeling. If you take a nutrition class, you're going to probably spend a week or two talking about how to read labels. But you see, you're given the total fat and the saturated fat. Now, some people get confused here. Well, the total fat, the USDA says that 10 grams is 15% of the amount of fat you should consume in a day. But whatever product this is contains 4 grams of saturated fat, and that 4 grams is 20% of the saturated fats you should have every day. Again, cholesterol, 30 milligrams, that's 10%, so that should tell you, if you do some simple math, you should never have more than 300 milligrams of cholesterol in your diet a day. Total carbohydrates, 44 grams, but then you've got dietary fiber, that's a carbohydrate. One of the things I was taught when I went through my diabetes education was that when you're counting carbohydrates, you can always deduct, there's a formula you use to deduct the dietary fiber. If you have the dietary fiber, you take it out, then that lowers the total carbohydrates. There's also something called, this label, particular label doesn't show it, but there's also something called sugar alcohols, which you can remove from the total carbohydrate as well. Protein, 21 grams. And then, again, vitamin D. Vitamin D is actually kind of an interesting vitamin, and we'll talk about this, and then you'll talk about it in your nutrition class if you take nutrition. Vitamin D is the only vitamin that we actually can make. If you sit out in the sun, you make vitamin D. Molecules are two or more chemically joined atoms. You see the sodium and the chlorine come together, water and methane covalent bonds. So you've got ionic bonds forming here. The electrons are changing places. The covalent bond, the electrons are being shared. Quick overview of what we've looked at today. Dehydration synthesis builds large molecules from small ones. You've got the glycerol. You've got the three independent fatty acids come together to form the triglyceride with water being removed. Cells can build polymers out of monomers. Again, the single monomer here, the single monomer here. Combine them. Again, both cases, you have water being lost. Carbohydrate. This is a long, each little green hexagon is the monomer. All together, they are referred to as the polymer. The protein, again, this is the monomer structure here. This would be a monomer. This would be a monomer. Put them together to form polymers. And then here, the monomer structure of the nucleic acid and the polymer that is whichever one of the nucleic acids that is. So, overview of this chapter. Again, we started with atoms in chapter 1, to proteins, to neutrons and electrons. Those atoms then form chemical bonds, which could be ionic, covalent, polar, nonpolar, showing the electronegativity. You can have hydrogen bonds. Chemical bonds form together to make molecules. You can have organic molecules if they contain carbon and hydrogen. Water. The molecules of water stick together by hydrogen bonds. And then you have all of these different other structures being made organically. You've got the carbohydrates, the proteins, the lipids, and the nucleic acids. Questions? If you have questions, please contact me either by my trussell@uaptc.edu email, or contact me through the message center under the course contacts in Blackboard. I don't hear any questions. Of course I don't hear any questions. There's no one in the room with me. Talk to you later. Russell out.